In 2020, a team of researchers did something that had never been done before at that scale. They collected genetic data from more than 50,000 people across 3 continents, West Africa, the Americas, and Western Europe, and they ran it through one of the most comprehensive genomic analyses ever attempted. What they found did not just add a footnote to science, it rewrote the genetic story of an entire people. Black Americans carry a combination of DNA that exists nowhere else on the planet. Not in Africa, not in Europe, not in Asia, nowhere. And the reason why is one of the most haunting, complicated, and ultimately remarkable stories in human genetics. Before we dive in, tell us where you are watching from and subscribe. Let's start with something that sounds simple but isn't. When scientists talk about the DNA of a population, they are not talking about eye color or height. They are talking about millions of tiny variations scattered across 3 billion base pairs of genetic code. Variations that tell the story of where a group of people came from, what diseases they survived, what environments shaped them, and what happened to them across hundreds of generations. And if you zoom in on the genome of Black Americans specifically, what you see is something geneticists describe as unprecedented, a tri-continental admixture, a blending of West African, Western European, and in many cases Native American ancestry, forged not through natural migration or peaceful intermingling, but through one of the most violent forced relocations in all of human history. Now, every population on Earth has a unique genetic story. The Ashkenazi Jewish population has one, the Icelandic population has one, the Han Chinese have one. But what makes the Black American genetic story different, what makes it singular, is the combination of factors that produced it: the scale, the violence, the geographic compression, the multi-generational forced mixing. There is no comparable event in the genetic record of any other modern population. To understand what makes Black American DNA unique, you first have to understand how ancient and extraordinary African genetics are. The story does not start in America. It starts on a continent that contains more genetic diversity than the rest of the world combined. Here is something that will stop you in your tracks. If you took every human being alive on Earth, all 8 billion of us, and split us into 2 groups, Group A being all Africans and Group B being everyone else, the genetic diversity inside Group A would be greater than the genetic diversity between the 2 groups combined. Read that again. Africa does not just have diverse people. Africa has more genetic variation within its borders than every other continent put together. This is because humans have lived in Africa for approximately 300,000 years. Every other population on Earth, Europeans, Asians, Native Americans, Pacific Islanders, descends from a relatively small group of Africans who left the continent between 60 and 70,000 years ago. Think of it this way. Imagine a jar with 1,000 different colored marbles. That is Africa. Now someone reaches in, grabs a handful of maybe 50 and walks out the door. The handful carries some of Africa's colors, but not all of them. Not even close. That handful became the ancestor of everyone outside Africa. This is what scientists call the founder effect. It means all non-African populations are, in a very real sense, a genetic subset of Africa. Europeans carry African DNA. Asians carry African DNA. All human DNA traces back to Africa. But here is where Black Americans enter the picture in a way that is completely scientifically singular. Subscribe to this channel if you want to keep learning stories like this. We cover genetics, ancient history, and the real science of human origins every week. Hit the button now. Between 1525 and 1866, an estimated 12.5 million people were taken from their homes along the coast of West and Central Africa and forced onto ships crossing the Atlantic. It was the largest forced migration in recorded history. Of those 12.5 million, only approximately 10.7 million survived the crossing. Almost 2 million people died in the holds of ships before they ever reached land. Those who survived arrived in the Americas carrying something no one at the time could have named or measured: the genetic heritage of dozens of distinct West and Central African ethnic groups: Yoruba, Igbo, Akan, Mandinka, Wolof, Fon, Senegambians, Congolese. Across nearly 3 centuries, enslaved Africans from what are now Nigeria, Angola, the Democratic Republic of Congo, Ghana, Senegal, Sierra Leone, Cameroon, and Benin were brought to a new continent. And when they arrived, something happened to their DNA that had never happened before, and has never happened since in quite the same way. They were forced to live alongside, and in many horrific cases forced to reproduce with European colonizers. Their genes began to mix, not voluntarily, not peacefully, but the mixing happened on a massive, multi-generational scale. And what that produced was a new genetic population, one that did not exist anywhere else on Earth. 2014, researchers published a landmark study in the American Journal of Human Genetics, analyzing the DNA of nearly 160,000 people across the United States. When they isolated Black American genomes, they found that the average African American carries approximately 73% African ancestry, 24% European ancestry, and 0.8% Native American ancestry. Those numbers alone make Black American DNA unusual, but they are not what makes it truly singular. Here is what does. The 73% African component does not come from one part of Africa. It does not come from one ethnic group or one region. The Broad Institute at MIT and Harvard analyzed the DNA of 30,000 African Americans and found that the African portion of Black American genomes contains contributions from dozens of West and Central African populations that, in their homelands, would never have mixed with each other. The Igbo of Nigeria, the Yoruba of Benin, the Akan of Ghana, the Congolese of Central Africa, the Mandinka of Senegal, all present, all blended together in a way that exists nowhere in Africa itself. This is critical. If you go to Nigeria today, you will find Igbo and Yoruba DNA, but not blended with Congolese and Akan in the proportions found in Black Americans. The slave trade took people from an enormous geographic and ethnic range, brought them to one place, and their descendants became genetically distinct from anyone who stayed behind. The African diversity inside Black American DNA is, in a very real sense, a compressed archive of West African genetic history. And that archive exists nowhere else in the same form. But there's a second layer, and this is where the science gets not just extraordinary, but morally heavy. The 2020 study from 23andMe, one of the largest genetic analyses of African ancestry in the Americas ever conducted, confirmed one of the most brutal realities of slavery in cold numerical genomic terms. They looked at the X chromosome. Here's why it matters. Men have one X chromosome and one Y chromosome. Women have 2 X chromosomes. The X chromosome carries a record of female ancestors at a higher rate than the autosomes, the regular chromosomes. When scientists compared the X chromosome ancestry of Black Americans to their autosomal ancestry, they found a stark imbalance. African ancestry in Black Americans is elevated on the X chromosome. In plain English, African ancestors contributed more through female lineages than male. African women contributed to the modern gene pool at 1.5 times the rate of African men, even though men made up more than 60% of enslaved people transported across the Atlantic. The math only works one way. African men died in the holds of ships, in fields, from disease and violence. They were killed before they could have children. African women, who survived at higher rates, were systematically raped by European men. That pattern was so consistent and widespread that it left a permanent, measurable signal in the DNA of every Black American alive today. The European ancestry in Black Americans skews male. European men contributed to the gene pool at 3 times the rate of European women. There is no other explanation. It is the genetic signature of centuries of sexual violence. Population geneticist Stephen Micheletti of 23andMe put it plainly. The mistreatment of people with African ancestry shaped the current genetic landscape of African ancestry in the Americas. That shaping is permanent, written in the genome of tens of millions of people. Now here is where things get more scientifically remarkable, because the slave trade did not just blend populations, it concentrated something, and that something is still being studied today. In 2010, geneticists discovered variants in a gene called APOL1. The variants, called G1 and G2, are found almost exclusively in people with recent West African ancestry. Approximately 50% of Black Americans carry at least one variant, and roughly 13% carry two. These variants did not arise randomly. They arose as a survival mechanism. Somewhere in West Africa, thousands of years ago, a parasite called Trypanosoma brucei, which causes African sleeping sickness, was killing people in enormous numbers. The APOL1 G1 and G2 variants gave carriers a powerful immune advantage. People who had them survived. Over generations, the variants became common. This is natural selection doing exactly what it is designed to do. But there is always a cost. In the United States, where there is no African sleeping sickness, those same APOL1 variants are associated with dramatically elevated rates of kidney disease. Black Americans develop kidney failure at approximately 4 to 5 times the rate of Americans of European descent. Part of the answer is written in the genome, in a variant that helped ancestors survive a deadly parasite, now operating in a completely different environment. This is not a flaw. This is what all genetic adaptations look like across time. Sickle cell trait follows the same logic, protective against malaria in African environments, carrying risk where malaria does not exist. The genome does not know where you are going. It only knows where you have been. And the APOL1 story is just one piece of a much larger picture. Black Americans carry genetic variants shaped by thousands of years of African evolution, variants extraordinarily rare or absent in every other population on Earth. They are not defects. They are the legacy of survival in a specific environment, now operating in a completely different one. There is a third layer, and it has to do with a concept called the bottleneck. In 2023, Harvard geneticist David Reich, one of the world's foremost experts in ancient DNA, noted something striking following a study of enslaved African Americans buried at the Catoctin Furnace Ironworks in Maryland. The study, led by Harvard, the Smithsonian Institution, and 23andMe, analyzed DNA from 27 individuals buried at that site and compared it to 9 million people in the 23andMe database. That small group of enslaved people had tens of thousands of living relatives and descendants alive in America today. Reich noted the pattern is suggestive of a genetic bottleneck, not unlike what happened to Ashkenazi Jews in Europe, where a small founding population expanded dramatically. Slavery's brutal conditions, the high mortality, the separation of families, the geographic concentration of enslaved people may have narrowed the genetic pool in ways scientists are still mapping. The people who survived passed their DNA forward through conditions that should have been genetically catastrophic. The Middle Passage, disease, starvation, violence, each was a filter. The people who made it through, who lived, who had children, who persisted across generations, left behind a genetic legacy specific to survival under conditions no other population has faced in quite the same configuration. This is not metaphor. It is literally written in the genome. Now you might be wondering, if most Black Americans trace their ancestry to Angola and the Democratic Republic of Congo, the largest source of enslaved people, why does the DNA show so much Nigerian ancestry? This is one of the most fascinating discrepancies the study uncovered, and it perfectly illustrates why genetics can tell us things written history cannot. Historical shipping records, the actual manifests of slave ships, show the majority of enslaved people sent to the United States came from the Congo-Angola region. You would expect Black American DNA to reflect that clearly. It does not. The genetic data shows a significant overrepresentation of Nigerian ancestry, specifically Yoruba and Igbo lineages from southeastern and southwestern Nigeria. The researchers were puzzled. Then they consulted historians, who explained the internal trade within the Americas. Enslaved Nigerians were frequently taken first to the British Caribbean, to Jamaica, Barbados, and other islands, and then traded again into the United States. The shipping records documented only the first leg of the journey. The DNA captured all of it. This is genetics solving a historical mystery that written records could not resolve alone. The bones of the story were there in the archives. The genome filled in the rest. And here is something that I want you to sit with. The Broad Institute at MIT and Harvard, while mapping the recombination hotspots of African American DNA, identified something that had never been found in any other population on Earth: a novel 17-base DNA motif, a specific sequence of genetic code present in people of West African ancestry and absent in every non-African population ever studied. 17 base pairs, unique to the African ancestral lineage. This is not a mutation. It is a feature of the genome, a signal of how DNA reshuffles itself across generations that only exists in this ancestral line. It has been in the human genome for potentially hundreds of thousands of years. It survived the ice ages, the migrations out of Africa, and millennia of disease and famine. It was there when the first humans walked upright. It survived the Middle Passage, and it is present in tens of millions of Black Americans today. So what does all of this mean? It means Black Americans don't just have a culture unlike any other. They don't just have a history unlike any other. They have a genome unlike any other. The specific combination of West African genetic diversity, drawn from dozens of ethnic groups across thousands of miles of coastline, fused with European admixture through centuries of coerced and violent mixing, shaped by survival bottlenecks and carrying ancient immune variants forged against deadly parasites, has never existed before in human history. It did not exist before the slave trade. It could not have. The conditions that produced it were historically unique. It was not designed. It was not chosen. It was imposed by one of the most horrific institutions ever created by human beings. But it happened, and the people who descended from it are the product of something genetically unprecedented. Geneticist Simon Gravel at McGill University has spent years modeling the admixture history of African Americans. He described it this way: the African American genome captures the genetic diversity of an entire continent, compressed into a single admixed population that then survived conditions that killed millions of others. What you carry in your DNA is not just ancestry. It is the record of survival. 48 distinct ethno-linguistic groups, 12.5 million people forced onto ships, 300 years of systematic violence. And from all of that, this people, this genome, this living archive of West African genetic history exists nowhere else on Earth. There's one more layer, and it has real consequences today. African Americans have significantly more genetic variation in their genomes than European ancestry populations. The West African populations at the core of Black American ancestry are among the most genetically diverse on the planet, and the admixture process created new combinations of variation that had never existed before. What this means for medicine is enormous and largely unresolved. The variants that predict disease risk, drug response, and cancer susceptibility are distributed differently in African American genomes than in European American genomes. Many variants common in Black Americans are rare or absent in the populations most genetic research has been conducted on. For decades, genome-wide association studies, the studies that identify which genes are linked to which diseases, were conducted overwhelmingly on populations of European ancestry. That means the genetic risk maps doctors rely on were drawn from the wrong population. The NIH has spent years trying to correct this through the All of Us research program, the African diaspora population cohort, and dozens of studies specifically designed to capture African genetic diversity. But the gap is significant, and the work is ongoing. This matters in emergency rooms, in cancer treatments, in kidney disease management, in cardiovascular risk assessment and drug dosing. The genome scientists built their tools on is not the same as the genome tens of millions of Black Americans carry. It is distinct, not worse, not damaged, but different in ways medicine is only now beginning to fully understand and account for. The people who survived the transatlantic slave trade left behind something that cannot be erased. It was written into every cell of the genome they passed to their children and their children's children, a genetic archive of an entire continent's ancient diversity, fused with the history of another continent, forged through conditions that should have been genetically devastating, and yet it persisted, generation after generation, through Jim Crow and the Great Migration. In every upheaval that followed, the genome kept passing itself forward. The genome does not know history. It does not grieve. It does not remember the way people remember. It just carries forward the record of who survived and who reproduced. And what it carried forward from the holds of those ships, from the rice plantations and tobacco fields and ironworks of colonial America, is a genome no scientist in the world has ever seen anywhere else on Earth. Not before, not since. A genome that is the direct biological consequence of one of the most violent chapters in human history, and at the same time, one of the most remarkable examples of genetic survival the human species has ever produced. That is not comfort, but it is truth. Sometimes the truth carries a weight of its own that is worth sitting with. This is the kind of story genomics keeps unlocking, stories that history recorded imperfectly or did not record at all, but that the genome holds on to forever. Science is only beginning to read them. If this video made you see something differently, share it. These are stories that deserve to travel.